Welcome back. Assalamu alaikum dear students. Today we are going to discuss vitamin A in detail. So let's go straight into the topic. This lecture has already been discussed in Urdu and uploaded in my first channel, Dr Hadi Sultan Academy. So those students who say we can't get English, please visit that channel first. Okay, in today's video we will be teaching you the chemistry of vitamin A, different structures of vitamin A, how vitamin A is absorbed and how it is transported, plus the rough function of vitamin A. Its main function in the vision, the visual cycle, will be discussed in the next lecture. But today we will cover only those things which are already on the whiteboard. Chemistry, history, transport, absorption, etc. Let me tell you something about the history of vitamin A. In the past, Hippocrates used to give the ox liver, of course mixed with honey, sweet honey, to those people, to those patients who were unable to see at the night time. Means they were suffering from the disease called night blindness. So the ox liver, ox liver, when prescribed to those patients, it brought a revolution in their eyes function. Means the disease just gone away by giving this ox liver. Nowadays, when we realize that what was present in the ox liver, that when we gave it to the patient became okay. So scientifically now it is proved that in the ox liver, vitamin A is present in large amount. So that was the history of vitamin A. Okay. Now, it is really important to know that vitamin A is present both in animal origin as well as from plant origin. In animal, you can call it just vitamin A, okay, or some kind of ester form of vitamin A. Simply, there is no special name for it. But in case of plant, vitamin A has got special name. That is called beta carotene. Here, beta carotene. So what is the definition of beta carotene? Beta carotene is the name of vitamin A which is present in plant. I would definitely discuss the structure of beta carotene with you people as well. But remember, carotene comes from carrot, and you know, people they love eating carrots, and every common person say that it helps in vision. Of course, carotene. Now, retinoids. What are retinoids? Of course, you can write what I say. Vitamin A may be naturally available in plant and animals, as I already discussed, or it may be synthesized in the lab, right? So both synthetic as well as natural forms of vitamin A are collectively called retinoids. Next, vitamins. Basically, vitamin A is not a single structure, just like water has formula H2O, single structure. Glucose has C6H12O6. Carbon dioxide, CO2, single structure. No. Vitamin A has closely related different structures, different forms of vitamin A. So you can't say vitamin A is just a single structure. Vitamin A is available in 3 different structures. These 3 structures are quite similar with each other, but there is slight difference exists in between them. Let me discuss this. Okay, these three different forms of vitamin A, they are called vitamers. So these three forms, their name are retinol, retinoic acid. Sorry, I am just using this kind of expression just to make sure that there is difference between the retinol, and retinoic acid. Fine. If you want to get the idea of this, I have already gave a detailed description of retinol, retinoic acid, even beta carotene in my one video lecture where I have discussed this, because I knew that when it comes to the lecture I have to revise it again, and it will take a lot of time. So that's why, in order to save the time, I have already recorded a video for this. Anyway, in the structure of vitamin A, you will find two things, two components. One is just a benzene-like structure, which is not benzene but looks like benzene, because there is only one double bond is there. 3 methyl groups are there. This structure is called beta ionione, and there is a long side chain, and this side chain contain 4 double bonds. And don't worry, just memorize it. If you want to write, so you can just go up and down 9 times. 1 2 3 1 2 3 4 5 6 7 8 9. So it's a nice side chain. Double bond here, you will write one double bond here while you coming from top to the bottom downside, then you will leave it, and then you will add another double bond here, and then you will leave this line, but you will add double bond here. You will leave this line and add double bond here. So a total of 4 double bonds you are supposed to add. Remember, these are double bonds, and in between these two there are carbon. Here is carbon. Each corner represents. You people are no more the students of matric level or FSC level. Even FSC level students, they knew. Yes, they knew that each corner actually represents a carbon. So we don't write this. So this is your side chain. And now there are 2 methyl groups. One CH3 methyl group is attached here. You will leave this one, and you will put one CH3 here. Then you will leave this one, and then you will add one CH3 here. Now this side chain, it is made up of small units called isoprene. And what is the structure of isoprene? Isoprene consists of 5 carbon molecule. Carbon number 1 2 3 4 5. There is 1 methyl, 4 carbon, 2 double bonds. Whenever you see such kind of molecule, this is always called isoprene. Isoprene, there's nothing special with this. This is just a formula, like carbon dioxide, CO2 is a formula. So isoprene is a formula. However, I would like to tell you that isoprene belongs to the lipid. It is not protein, it's not carbohydrate, it belongs to the lipid. So isoprene. There are 2 isoprene units that combine to give us the side chain. So in this side chain there are 2 isoprene units. You can say that from here, this one, look, there is CH3. There is a CH3. 2 double bonds, 2 double bonds. So this is 1 isoprene unit. Luckily, interestingly, how I could skip this point, maybe if it takes a long time, I have to mention it. And this one is our second isoprene unit. Fine. So there are 2 isoprene units. Now, regarding retinol, if you add here, if you add CH2OH, it will become retinol, because OH is there, retinol. Look, just a small difference is there in the general structure of vitamin. And if you erase this one, you write C double bond O, and there is H. This is of course aldehyde functional group, but the whole structure will be changed. The name of that structure will be called now retinol. And if you add COOH, so lovely group, functional group, carboxyl group. Now it will become what? Retinoic acid finally. So you can read. This was the chemistry of vitamin A. However, one thing left, beta carotene. Beta carotene has similar structure just like this vitamin A. However, there are two vitamin A structure combined. Two. That means this is one vitamin A structure, ring, side chain, ring and side chain. If two vitamin A structure are combined by the help of one double bond, look at this, there is no CH2OH, there is no C double bond OH, there is no COH. However, here you can just add two double bonds here. Now this structure now is called beta carotene. And remember, this beta carotene can be cleaved, means can be broken down by the help of enzyme, and that enzyme is called dioxygenase. Please remember the name dioxygenase. Beta carotene, you can call it beta carotene. CAR is just shortcut form of carotene. Beta carotene dioxygenase enzyme can break this structure into vitamin A, two vitamin A. Of course, when this structure is broken down, you will get two vitamin A. However, it will be retinol when it is broken down. It will be retinol. Okay, fine. Now come to the absorption. Regarding absorption, definitely you will take vitamin from the diet. What will you take? That is your diet. So when you take something, food that contains vitamin A, you can take them either in beta carotene form. I already discussed, means if you are taking plant, some vegetable, so definitely you are taking vitamin A in the form of beta carotene. Beta carotene. It is also called provitamin A. Or in case of animal, if you are eating the ox liver, so you are also taking vitamin A, but this time vitamin A is called retinyl esters. Don't worry, retina word is still there. Okay, retina loves. So retinal is still there. Ester comes. So what is ester? Don't worry. I will tell you vitamin A plus fatty acid. Whenever these two are combined, this is called ester. Fine. So vitamin A plus fatty acid is called retinyl ester. And do you know which vitamin A is here? All form or all form or oic? This is all form. Remember, retinol, vitamin A, which vitamin? Retinol. So retinol plus fatty acid, it will give us retinyl esters. Okay, fine. So when you take the diet, beta, of course, whatever you eat, it will come into your intestine. This is your intestine. Inside the intestine, there are large number of cells at the inner wall of the intestine. Large number of cells are there. Okay, but just one cell, if I take just one cell here, and this is the diagram of just one single intestinal cell, beta carotene will be absorbed directly into that intestinal cell. Of course, from the lumen, this is called lumen, L-U-M-E-N, lumen of your intestine. From the lumen of the intestine, it will absorb into the cell, and this is cell. Okay, so beta carotene is here, beta carotene. Beta carotene will be converted into retinol by the help of an enzyme, and this enzyme is called beta carotene dioxygenase. Thanks God, I already discussed that. So this enzyme will give us retinol. Of course, two retinol, because beta carotene consists of two vitamin A. So retinol, it will be retinol. Remember, that retinol will be reduced. Retinol will be reduced into retinol. So simple reduction will be there. This retinol is now combined with fatty acid. I already told you, whenever retinol combines with fatty acid, it will give us ester. So here is one fatty acid inside your intestinal cell. This fatty acid will combine with retinol and we will get retinyl esters. Just like retinyl esters, its shortcut form is REES, retinyl ester. So we got this retinyl ester. Finally, the drama is over now inside the cell. This retinyl ester is actually the protected form of for the vitamin A. It is very stabilized now, you can see, but it is unable to travel in the blood. It's very difficult for it. Now we need something more. We need to add something more with this retinyl ester so that it can safely travel inside blood. And what is this structure? That is called chylomicron. Chylomicron, I have a video lecture on chylomicron. Please watch that video on what is chylomicron. However, in today's lecture, I would like to tell you that chylomicron is the combination of two things, that is lipid and protein. So lipid protein combination is called chylomicron. I know this definition is not enough to tell you about chylomicron, but still this is just a shortcut to have some idea. So the chylomicron is a very beautiful structure. This is a spherical structure that it usually carry lipids inside body from one place to another, you know, like LDL, VLDL, good cholesterol, bad cholesterol, etc. all of these. So chylomicron will take this vitamin. I said, okay, come on, vitamin A, I will take you inside blood, from blood to the liver. Remember, what is absorption? Absorption is this transfer of vitamin A from intestinal cells into the blood. This is called absorption. Once the vitamin A has arrived in your blood, you can call it absorption. So now there is transport. The vitamin has to transport from one place to another. So initially in the blood, the vitamin A is transferred to the liver. This is your liver. In the liver, vitamin A will be stored. Now, of course, we have chylomicron, but in the liver, chylomicron is removed. One. In the liver, that esterified form, it is also removed in the liver. In the liver, you have retinol. Finally, this retinol is arrived here. And don't worry, what I have mentioned here, all trans. What is this trans? Don't worry. Actually, this trans is representing the isomer of vitamin A. You know, in organic chemistry, different compounds have isomer, cis and trans isomer. You might have heard this word, cis and trans isomer, but if not, I can tell you that when there is double bond between two carbon, and the molecule is organized in such a way that one CH3 is at the top and other CH3 is at the bottom. This kind of isomer, which involves a double bond and CH3 at the opposite side, is called cis isomer. Trans isomer. Trans. And if the CH3, both the CH3 are on the same side, this is called cis. Actually, in our vitamin A, there are 4 double bonds. Not only one, 4 double bonds. So all of these double bonds are presenting us the trans form. Which form? The trans form. So that's why we call it all trans retinol. So this all trans retinol will arrive in your liver, and that retinol is either stored in the form of palmitate. Palmitic acid is another fatty acid that is attached with the vitamin A, and it is stored as such when it is combined with palmitic acid. Fine. So this is enough. Just it will be stored. But remember, vitamin A cannot be stored as such. Vitamin A retinol, a fatty acid must combine with vitamin A, and that is palmitic acid, palmitoyl esters. So it is stored. I did not mention here that what happened, but I have just expressed. From the liver, the vitamin A has to move to the body parts where it can deliver its function. Means vitamin has to travel in the blood from the liver to our eye. From the liver to our eye, definitely there is blood. In blood, vitamin A has to travel and go to the eye. So in the eye, vitamin A will perform its function and will happen in vision. Of course, that is wild cycle. You please remember this thing, that today in today's lecture we cannot discuss this wild cycle because it requires an explanation that I am planning to record in another video. Means how vitamin A helps in vision. But other functions, vitamin A has also other function as well. Means growth and differentiation. Your body contains cells. In the beginning, cells don't have a function. Once cells get some particular function, you call it differentiation. So inside our body, cells need to be differentiated. So vitamin A has a role in its cell differentiation. How I will discuss this one, that how the mechanism of differentiation. And your skin, vitamin A helps in providing you a smooth skin as well. So now we need to take vitamin A from the liver and it will go to the target cell. From the liver, vitamin A can be mobilized, means detached, by the help of zinc. When zinc comes, zinc will knock the door of liver, please give us some of the vitamin you have stored, but give some of the vitamin because there is need of it, body require it. So the zinc, without zinc, the liver cannot allow the vitamin to release itself. So zinc is necessary. So that is why the role of zinc in vitamin A. The role of zinc in vitamin A. If some students they don't know, they just use cheating, asking from the colleagues here and there. Or just 3 lines definition. Role of zinc is this and that. Okay, so once the retinol again enters the blood, again in blood, as I already told you that vitamin A as such cannot travel in blood. In that case, chylomicron helped vitamin A. Okay, come on, I will take you to the target. But from the liver, chylomicron cannot do this job. Chylomicron say, I'm sorry, I can't. There is another protein inside your blood, inside your plasma. This is called retinol binding protein, RBP. So special protein. Please note the name of this protein, RBP. RBP combines with retinol. This complex is now there, vitamin A and RBP. These two will now move towards the target cell in blood. Once it reaches to the target cell, RBP, retinol binding protein, it will be just removed because this RBP has played its role. Now vitamin A needs to enter the cell. Here it requires another protein that is called retinol binding protein, again RBP. The difference is this one is plasma RBP, but this one is the cellular RBP, inside the cell. Now this RBP will definitely welcome the incoming vitamin and will say, hello vitamin A, we are welcoming you here to our special cell. This RBP will take vitamin A and it will guide the vitamin straight to the DNA. Means even to the nucleus. There are a lot of guards, securities there around the nucleus. So by the help of this RBP, the vitamin A can be safely delivered into the DNA, into the nucleus where DNA is present. And you know, DNA is such a tremendous controlling molecule inside our nucleus. So when the vitamin A attaches at the DNA at a specific site, vitamin A, which form of vitamin A? This is really something to know. Either retinol, or retinoic acid. So keep in mind, this is a retinoic acid. So it's attached to the DNA and it will order, it will release the order, it will issue an order that I need messenger RNA. So messenger RNA will be released from the DNA, will be synthesized from the DNA, and that messenger RNA will definitely make a protein. You know very well that messenger RNA will provide us protein. This protein will go and help in the differentiation. So that was the whole story of vitamin A. I hope you enjoyed a lot. I will just take aside, you can have a screenshot of all what we discussed, the chemistry of vitamin A, that there are 3 forms of vitamin A called vitamins. One is all, and oic acid. These 3 are very similar with each other, identical. The difference is only in the functional group. Okay, and then we also discussed beta carotene. We also discussed that dioxygenase enzyme can break this into 2 vitamin molecule. And then we discussed the absorption and transport. Absorption we have done that from your diet, vitamin A can be either in the beta carotene form or in the retinal ester, whatever the case is. If it is beta carotene will come into the cell and it will be hydrolyzed by the enzyme into retinol and reduced into retinol, and then fatty acid combined to give us retinal ester. In case of retinal esters, if you are taking it from the animal origin, of course, from this retinal ester, the fatty acid will be removed. The fatty acid will be removed, and retinol will be will enter in that cell. So this retinol represents beta carotene as well as this retinal ester. Whatever the case is, both retinal ester and beta carotene, they will enter the intestinal cell and ultimately they will become retinol. Okay, and then when it is esterified in ester form, it will combine with chylomicron, and in the form of chylomicron it will travel in the blood, reaches to the liver. In liver it is either stored by the help of palmitic acid, or it is transported to the other body parts where it can deliver its function. So we saw that it is moving to the eye where it can help, where it can participate in the vision cycle, ward cycle, or it is transported to the target cell. Here once again zinc is helping the vitamin A in its mobilization, and then RBP a retinol binding protein, it is attaching with the vitamin, and it will guide the vitamin A to the target cell. Inside the target cell, another RBP molecule will receive the vitamin, and it will guide the vitamin straight into the nucleus and will attach the DNA. At the DNA, when it attaches, then messenger RNA is synthesized from the DNA, and that messenger RNA will give us a protein, and that protein will help in the differentiation. That was all the lecture of vitamin A. I hope you got it. If you got it, give us a like, and please share this channel with your colleagues. And as usual, remember us in your prayers. Thank you. Allah Hafiz.